Welcome back. And like I've mentioned before in our study of polygenic inheritance, most of the traits in humans that are most obvious to us and most interesting to us tend to be polygenic. In fact, most human traits are polygenic. But we needed to start out last semester with single gene inheritance just so that we could understand how alleles work, how gene pairs work. But keep in mind, again, we're talking about many gene pairs working together in an additive fashion. I don't know if you remember in first year biology learning about genetics, you probably used a very simplistic model to describe the inheritance of eye color. You probably learned something like this. Capital B is the allele that causes brown eyes. Small b is the allele that causes blue eyes. And so you did your Punnett squares, big B, little b, crossed with, let's say, little b, and you would begin to see that, gosh, some individuals are going to have brown eyes, some are going to have blue eyes, but maybe you were an inquisitive freshman and you thought, well, how about all the other eye colors in the world? Well, knowing what you know now, you know more than you did as a ninth grader in first year biology. How would you explain how come there are so many different shades of eye color, not simply just brown and blue? How do you account for all the different shades of eye color? And I'm going to pause for a second and let you think about that, and then we'll come back and discuss the answer to that question. Okay, I hope you were thinking, well, because it's polygenic. Eye color is polygenic. It's controlled by more than just one pair of genes. Who knows? Maybe it's 5 pairs of genes all working together, but this can create all the different variations in eye color that we see from really dark brown at one end of the continuum to very, very light blue at the other extreme end, and all the different shades and colors in between. So eye color is polygenic, just like skin color, which we've talked about before. So now let's move on here. Let's switch gears a little bit and talk about some traits that are polygenic that are part of the construction process of the fetus. So, for example, the trait I'd like to look at is the closure of the neural tube. Remember that from a long, long time ago, last semester when we were learning about how the human embryo is put together. Remember we talked about how at 3 weeks after fertilization, a groove forms down the back of the embryo. And in this picture on the left-hand side of an embryo, actual photograph of a human embryo, we're looking at a neurula stage. That's where the neural tube forms. We're looking down on the back of the embryo here. And the embryo from head to tail in this photograph is about the size of a grain of rice, uncooked. And you can see the neural tube is closed up almost all the way except up here at the very head, top of the head. It's not quite closed yet. So that neural tube has zipped up, it's closed up to form this hollow tube running down the back of the embryo called the neural tube, which will eventually become the brain and spinal cord. If you look at the photograph on the right, you'll see a human embryo just a little bit past 3 weeks of embryo development. And you can see here that, in fact, the neural tube has completely closed up. So this is a normal embryo whose neural tube is closed up like it was supposed to close up. Now, that closure of that neural tube is under the control of many gene pairs working together, so that trait, the closure of the tube, is a polygenic trait. Now, sometimes, if you'll remember, it doesn't close completely like it's supposed to, and we end up with a condition like this called spina bifida. So this is a photo of a newborn baby with a very large cyst in the back, indicating the position where, when that child was a 3-week embryo, his neural tube failed to close up, in the lower to mid back region right there. And so during the course of embryo development, spinal fluid leaked out of that opening, collected underneath the skin, and formed this cyst on the back of that newborn baby. So this is called spina bifida, and this should bring back some memories of our unit way back last semester when we learned about embryo development. So keep in mind this closure of the tube is a polygenic trait. And if there's a problem, like in this photograph right here, we say that this is a multifactorial birth defect because the failure of that tube to close at that location along the spine, the failure of that tube to close could have been caused by some of those polygenes not working right, or it could have been caused by something harmful that mother was either exposed to in her environment, or some important nutrient that she was missing in her diet may have contributed to that, or it may have been it may have been the result of both, a combination of those environmental causes during the pregnancy and genes failing to work correctly. So we sometimes refer to this kind of a defect as multifactorial. Let's take a close-up look at a few more details concerning neural tube defects like spina bifida.
Spina bifida, or its most severe form, myelomeningocele, is the most common central nervous system or neurologic birth defect in the world.
Myelomeningocele is a defect in the spine. It's failure of the spinal cord to close.
The spinal cord, or the neural tube, as the embryologists say, forms very early in gestation, certainly before 8 weeks gestation. And spina bifida is thought to be a failure of the formation and closure of that neural tube, so that the spinal cord elements are exposed to the intrauterine environment.
It's a progressive process. It's a progressive injury, so that by the end of pregnancy, there's been direct traumatic injury to the spinal cord, and there's been injury from the amniotic fluid itself.
Because of changes in pressure and fluid flow, babies are also at risk for developing hydrocephalus,
which is a fluid buildup on the brain and which damages the developing brain.
Virtually every child with myelomeningocele or spina bifida has an Arnold-Chiari malformation on X-ray. And what it is that the back part of the brain, called the cerebellum, that has a lot to do with control of motor movements, is wedged down into your neck.
The cerebral spinal fluid that cushions the brain and cushions the spinal cord leaks out through the back through the open spina bifida defect. That leads to the back part of the brain getting sumped down into the upper part of the spinal canal. That in turn can block the circulation of cerebral spinal fluid in the brain, because the fluid is produced in the brain and then there's no way for it to circulate beyond that blockage point.
So if there's a blockage in the flow of that fluid, then the water builds up inside the ventricle. So you've probably heard the term water on the brain. The water isn't on the brain, it's inside the brain. If you don't treat that, then you end up with a massively enlarged head, or you die, or you end up with severe mental retardation.
So in order to minimize that ongoing damage of pressure from the fluid, the pediatric neurosurgeon places a shunt tube.
It's a simple device. This is a shunt. It's made out of silicone. This end goes through a hole in the back of the head into that water cavity and tunnels down under the skin and goes into the abdomen, which is able to absorb the fluid. It's not a cure. It treats the symptom. It doesn't treat the disease.
Once you have a shunt, there are multiple complications that can arise from it.
The shunt can become infected, which is a serious complication, because then the brain and the layers in the brain can become infected with meningitis and encephalitis. And with each bout of infection, that can affect the brain function.
And then, of course, there are mechanical problems. They clog, they break, They get to be too short. They migrate.
So it's not unusual for a child with spina bifida who has a shunt to require that shunt, and not only for life, but to have that shunt changed at least 5 or 6 times during the course of their life.
It's important for parents to understand the potential complications or ramifications of spina bifida after the diagnosis is made.
A child may experience difficulty with apnea, forgetting to breathe. They may also have breathing problems, choking, blue spells.
And in the worst case, you end up with a child that's on a ventilator with a tracheostomy and a tube into the stomach to eat. And that is a huge impact on your quality of life.
Spina bifida can cause paralysis in the lower extremities that can interfere with the baby's ability to walk, or they may even be wheelchair dependent.
They also have difficulty with their bowel and bladder function.
Most of these kids, when they become older, have to catheterize, or else they have to wear diapers.
They can develop significant curvatures of the spine because of instability, bony instabilities of the spine if they have a high lesion.
Because of the lack of innervation to the feet and the ankles, there can be the formation of club feet, or the medical term is talipes, as a consequence. So that requires orthopedic intervention after birth.
And then there are problems that can come on later on in life. It's not even necessarily done when you're a baby. You can get tethered cord, which means that the nerve tissue gets caught up in scar tissue, and you can lose function. So it's a lifelong issue of disability.
An important concept for us to consider is the concept of threshold effects. So when we're talking about a birth defect like spina bifida, it appears that the birth defect won't show up until there's a certain critical threshold number of faulty genes involved. So let's just assume, we don't know exactly how many genes work together to control the closure of the neural tube, but let's just say it's 20 gene pairs all working together, and in order for that neural tube to close up properly, those 20 gene pairs all have to be capital letters. Okay, so imagine capital A, capital A, capital B, capital B, capital C, and so on through 20 gene pairs. If one out of those 20 gene pairs isn't working right, there may not be any problem. The neural tube may close up like it's supposed to. If 2 out of those 20 gene pairs aren't working right, the neural tube might still close up normally like it's supposed to. If 3 out of those 20 gene pairs are not working right, we might still have a normal neural tube closure. But if maybe 4 out of those 20 gene pairs aren't working right, then the disorder may manifest itself. And it appears that the more faulty genes there are, the more severe the neural tube defect. So some neural tube defects are very mild. There might just be a tiny little hole at the base of the backbone that's hardly even obvious, maybe not even open. And you might have remembered the last semester we called this spina bifida occulta, where there isn't even an opening at birth, maybe just a little dimple down there. Or at the other extreme end of the continuum, it could be so severe that the entire spinal column is open at birth from the tailbone to the eyebrows, which you can imagine will result in a stillbirth. That child would just not make it at all. So there can be a range in severity there, which is another clue that tells us that this trait is polygenic. So when I think about threshold effects like that, it reminds me of when my wife and I were first married. My wife has always been the cultured one, and I was less cultured. I freely admit that. And I remember when we would first go to like a symphony concert. She's a musician, a very accomplished flute player, outstanding musician. She has a good ear. She's just really good at that. I'm not quite as good at that. Now, I was in choir, but I was not in orchestra. And so, whenever we would go to a symphony concert, the music could sound perfect to me because my ear is not as critical. I don't pick up on the mistakes among the musicians that maybe my wife might be able to hear. And so, if you think about a symphony orchestra, is kind of like all the musicians are all the genes responsible for a polygenic trait, like closure of the neural tube. And if, let's say, I don't know, let's say there are 30 musicians, if one of them messes up, the music might still sound pretty good. If two of them mess up, the music might still sound pretty good. If three of them mess up, then you might start to notice that the music is not as good as what it could be. And I remember going to a concert and I thought the music sounded great, but driving home, my wife was talking about how the flutes were a little bit off or something. So I didn't pick up on that. So in other words, our thresholds were different. And I don't know, that just kind of reminds me a little bit about the way genes work. Think of all these genes that are responsible for closing up the neural tube. They're sort of like musicians in an orchestra. And if a certain critical threshold number of them mess up, maybe this number right here, then you can see there's a threshold there where the music sounds bad. Or if we're going back to looking at how genes work to control the formation of the neural tube, if you have this where the red arrow is, if you have this critical number of threshold genes not working correctly, then the disease manifests itself. So if you'll look at this graph, on the x-axis, we're looking at genetic liability. So in other words, if you start at the origin of the x and y axes on this graph, as you go from the origin to the right along the x-axis, as you move to the right, we're looking at increasing numbers of inactive or mutated genes. And as you move from left to right, if you have an increasing number of inactive genes, there's no problem at all in the formation of the neural tube until you get to where the red arrow is. And when you have that critical number of inactive genes that are supposed to close the neural tube, then the child ends up with a neural tube defect of some kind. And to the right of the red arrow at the bottom of the graph, any increasing number of genes in excess of what is indicated at the right of the red arrow will result in ever increasing in severity neural tube defects. So let's look at what would happen then in a developing embryo if the unborn baby had, let's say, that number right there of abnormal genes responsible for making the neural tube close. Okay, so look at the position of the red arrow where it is right now on the slide. If the child had that particular number of inactive genes or abnormal genes, would that particular child end up with a neural tube defect? So I'm going to pause for a second, let you think about that, and then we'll come back and we'll talk about the answer to that question. Okay, so if you said that in this case right here, where the red arrow is, that this particular unborn baby would not experience a neural tube defect, you're absolutely right, because you can see the child does have quite a few inactive alleles. I mean, look how far we are from the XY origin there, but still not at the threshold level. So this child's neural tube would turn out to be totally normal. Now, remember I said closure of the neural tube is is a multifactorial trait. So not only are genes involved here, but environmental insults could cause a problem as well. There's some evidence that if mother has a very high fever at this critical period of embryo development, let's say 3 months, or not 3 months, 3 weeks after fertilization, a very high temperature due to a fever or hot tubs or something like that could interfere with the developing closure of that neural tube. That let's look at what would happen if during that time, if the baby has the number of inactive alleles represented by the red arrow, if during that critical time period mom's body temperature was increased due to fever or hot tubs or something, watch what those environmental insults could do to the threshold level. So as you can see, bad things in mom's environment that she's exposed to during that critical time of embryo development could, in effect, move that threshold level back to the left, and this child then would end up with a neural tube defect of some kind, because the bad things in her environment, the environmental insults, have in effect moved the threshold level back. With those environmental insults, high body temperature, a smaller number of inactive genes would still result in a neural tube defect. Now, let's look at another case. Let's say we have an unborn baby who is blessed with this number of inactive genes or abnormal genes. Look and see where the arrow is. Do you see where the arrow is? Okay, this shows a baby who happens to have very few inactive alleles or very few abnormal alleles that could potentially mess up the closure of the neural tube. So in this case, with very few abnormal genes involved, if mom's exposed to environmental insults of some kind, then you can see that the environmental insults would have in effect moved the threshold level back, but this baby has such a small number of abnormal genes to begin with that the baby still does not end up with a neural tube defect. The problem is, in any unborn baby, any developing embryo, we don't know how many genes the baby might have that would predispose the baby to having a neural tube defect of some kind. It is interesting, though, and I think this is very practical information for anybody and everybody. There's plenty of evidence now that if mom has enough or adequate folic acid in her diet, folic acid is in the B vitamin group. If mom has enough folic acid in her diet, that this can actually prevent neural tube defects. In other words, having an adequate amount of folic acid in mom's diet can, in effect, move that threshold line to the right. And so this is why women who are of childbearing age, especially women who are of childbearing age who are sexually active, are encouraged to be on folic acid vitamin supplements, just in case they get pregnant and they've got that protection. I know that last semester when we were talking about single gene traits, we talked about how certain ethnic groups have increased risk for certain single gene disorders. For example, sickle cell anemia being most common among people of African descent, cystic fibrosis most common of people from Western European countries, and Tay-Sachs disease most common among certain groups of Ashkenazi Jewish populations. It appears that neural tube defects are slightly, just a little bit more common among people whose ancestry goes back to Scotland, Ireland, and Wales. Now, if your ancestry goes back to those countries, that doesn't, of course, mean you have to freak out. It's just that it is a slight increase in frequency among people of those populations. And so, if you find yourself in that ethnic group and you decide you're going to have children someday, of course, you want to remember the news about folic acid and make sure you have folic acid supplements in your diet. So, spina bifida, again, failure of the neural tube to close up at just only 3 weeks after fertilization. This diagram right here at the bottom of the slide shows kind of a cross-section drawing of what happens here. And as you can see there, if the opening in the spinal column is large enough, you see the problem is spinal cord tissue could get extruded through that opening, where it ends up bundled up outside the body in this cyst. And if that cyst is somehow damaged or squashed somehow during birth and delivery, then that spinal cord tissue could be damaged, that spinal cord tissue that's extruded through that opening, resulting in paralysis or partial paralysis in the baby from that point down. Incidentally, it's kind of interesting, and you could Google this if you're interested to know more. There have been a fair number of surgeries done, prenatal surgeries, where surgery has actually been done on unborn babies to correct those kinds of neural tube defects and actually prevent the paralysis once the newborn baby gets here. Those are still fairly experimental, but there have been a few of them that have been done successfully. Here's another example of a multifactorial birth defect caused by a failure of tissues to close in the development of the early embryo. This one happens at 5 weeks after fertilization, cleft lip and palate, and it can appear to be fairly dramatic and very serious looking. But I want you to look at this little baby boy who was born with a cleft lip. After a couple surgeries, as he got a little bit older, and even a couple surgeries later as a young child here, showing a remarkable improvement in the child's appearance, and more importantly, the ability of the child to eat and for his teeth to develop properly. So when and how does this particular defect happen? It happens right here, 5 weeks after fertilization. We're looking face on at a human embryo 5 weeks after fertilization. At this stage, really doesn't look all that human, I know, but we're looking very early here. The embryo is probably about the size of maybe a ½ inch long, and on one side of the head, this would be the embryo's left side of the head, you've got an eye and a nostril on one sort of plate here that's going to form half of the face, and on the embryo's right side of the head, there's the other eye and the other nostril on the other side of the head, and those two plates have to come together. Kind of reminds me of plate tectonics in geology. Those two face plates have to grow together, and that growing together of those face plates is under the control of many genes working together. So again, it's a polygenic trait. If something interferes with that closure, either genes that aren't working the way they're supposed to, or environmental insults that mother's exposed to during the pregnancy, or a combination of the two, then the cleft lip and palate could manifest itself. And in that case, we'd call it polygenic because it's controlled by many genes working together. We also call it multifactorial because oftentimes we can't be sure. Was it genes not working the way they're supposed to? Or was it something mom was exposed to during the pregnancy? Or was it a combination of the two? I want you to take a look at this next video clip. Interesting video clip of a young lady who was born with a very severe cleft lip and palate. She's had numerous surgeries since then, and I think you'll find her story interesting.
Hi everyone. So this is my first video of my cleft lip and palate story. My name is Johanna, and it's taken me about a good 20 years to finally decide to share my story. I've seen other videos on YouTube, and they inspired me to, want to share my experiences and my stories. So for those of you who know me, you've probably been told like a good deal about me regarding this, because either you asked me or I felt like telling you because we've known each other for so long. For those of you who don't know me, I hope this video helps gain more awareness towards other people like me, and I hope that it helps you understand me as an individual. So, yeah, here goes. Cleft lip and palate. It is a birth defect. About 1 in every 700 children are born with cleft lip and palate. I think not just in the US, I think it's around the world. I heard that kids mostly like in areas around Indonesia, it's more prominent there. Not too sure about that fact, but yeah. So, cleft. Cleft means a gap or an opening. So when I was born, I specifically have a bilateral cleft lip and palate. That's why I have these 2 scars here. Meaning that there were openings. It wasn't fully fused together here, so they had to close it. Same with my palate. Your palate is the roof of your mouth, inside your mouth. And that connects to your nose. So it was hard, definitely as a child at first, before they did any surgery. Since it's 1 in every 700 children, it's fairly common. I've actually seen, a good number of people just around town and stuff with cleft lip, and I'm like, hey, we are the same. So, the complications that come with being born with cleft lip and palate, 1, is problems with feeding. When I was born, when I was a baby, they had to feed me milk through a dropper, so I couldn't really breastfeed. It was hard to suck, I guess. I couldn't breastfeed, and I couldn't drink from a bottle, so they had to feed me with a dropper. Another complication is ear disease. Since your nose and your mouth and ears are all kind of connected together, there will be complications. If there's openings in your mouth, you can easily, get stuff in your nose. When I was younger, I would get frequent ear infections. I think it was in my left ear. I just took kind of antibiotics for it, I guess. It was very frequent. I remember going to the doctor a lot when I was younger because of the same problem, ear infection. So, yeah, that was not fun. Another thing is speech. When there's openings or, in your nose and in your mouth, air is going to flow differently throughout, and what you hear is going to be different in your voice. So through elementary school, first to 4th grade or 1st to 3rd grade, I took speech therapy. It helped a lot. I thought it was like a lot of fun. I didn't really like realize why I was even in speech therapy, but it helped a lot. So that's probably gonna come with cleft lip and palate. Um, and socialization. Me personally, I never really had a big problem socializing. When I was younger, I was pretty shameless. I did not care, and it's pretty funny because like now it's like, oh, I care now. So I've had a total of about 10 surgeries. Not really sure if I've had 10 because I lost count, to be honest. This is excluding the one in the Philippines after I was born. All my surgeries then have been done here in Chicagoland area at Shriners Children's Hospital. So that is located in Oak Park, Chicago. I could give a link to the website for those of you who are interested or want to know more about Shriners Children's Hospital. They treat children with not just cleft lip and palate, but with orthopedic and neuromusculoskeletal conditions and spinal cord injuries. They have other branches around the country. They have hospitals from Canada to Mexico, so you can search that in their website. So, from my personal experiences in Chicago, as a kid, they definitely knew what to do and what to say to make me feel comfortable and more at ease with myself and like being in the hospital for surgery. It's definitely an unnerving feeling as a kid and having to go through like so many surgeries and you know, for the families too. So they definitely did a good job to put like all of us at ease and make us feel at home and very comfortable. I already said that I'll put a website and a link in my info. With that said, I'll explain more in detail about, you know, the nitty-gritty stuff in my next videos, because I'm sure like all of you really want to know more about my background, treatment, like more detail on like what kind of surgeries I went through and all that good stuff. So any questions for me, please leave a comment, subscribe, and thank you for watching. Okay, bye guys. And girls. Okay, bye everyone.
So anyway, I hope you found that video clip to be interesting. You've probably seen people before who have had, you can tell by looking at them that they were born with a cleft lip or a cleft lip and palate. And like most uh, multifactorial birth defects, there can be a range in severity. Some children born with cleft lip, it's very mild, might only require a few stitches. Some children born with cleft lip, it's at the other very severe end of the continuum. Remember our bell curve, where some are so severe that they require multiple surgeries as they're growing up. There's another very common multifactorial birth defect that we haven't even talked about, although we did look at it some last semester, congenital heart defects. And again, you can see a continuum with congenital heart defects. Some are very mild, heart murmurs that maybe a child may even outgrow, and some at the other extreme end of the continuum can be very severe, life-threatening. Now, there are some uh, unfortunate conditions that affect people uh, that happen much later in life. So let's switch our attention from what's happening during embryo development to what's happening later in life. And the example that I'm thinking of would be many cancers, for example, occur later in life. And if you'll remember from some of our discussions earlier in the semester, I mentioned that most cancers are not hereditary. In other words, they don't run in families, but all cancers are genetic. In other words, all cancers result from some sequence of DNA that mutates, goes haywire, and causes the cell that contains that particular sequence DNA to go haywire and to start dividing uncontrollably and resulting in a clump of those cells, which is called a tumor. So this is how all cancers develop. Um, the example of cancer that I want to talk about just for a moment, because it reminds me of a story of when I was a little kid, is that of lung cancer. And if you'll bear with me, we're almost finished here with this lecture, but I want to talk a little bit about a time when I was in the 5th grade, and we lived in Monticello, Indiana at the time, and it was 5th grade and summertime, and so a lot of times when I was in the 5th grade in the summertime, you would find me on my bicycle with maybe 3 or 4 of my buddies of the same age. We always kept a baseball mitt on the handlebar of our bicycles because you never knew when we might run into a pickup baseball game. But one of the things we liked to do, if we had collected enough money from well, we got a little bit of an allowance, but mostly as 5th graders, we made our money by riding around town and on the edge of town on our bikes looking for glass pop bottles that people had thrown out. And if we collected a certain number of glass pop bottles, we could take those to the A&P, the grocery store, and we could trade them in, and they would give us a little bit of cash for those. Not much, but a little bit. We could cash those in. And so we did that a lot. We went around, riding around looking for these pop bottles and so that we could trade them in for cash. And if we had enough cash, we'd go up to this small privately owned gas station, and we would go in there and maybe buy a candy bar or a bottle of pop or something like that. But there was this one old guy who sat outside the door to the entrance of the gas station where you could go in there and buy candy bars and stuff. And this old guy sat out there right next to the door, and he smoked like a chimney. Now, this is not actually a photograph of him. This is just a picture I found somewhere, but it reminds me of this guy. He was ancient. Now, I don't know how old he was. I think he was in his 80s, but he sat there right next to the door. And so, in order to go into the little gas station store to buy a candy bar or whatever, we had to walk past this guy. And we were scared to death of him. Remember, we're 5th graders. From on one of his legs, from the knee down, he had a wooden leg. And he would, he'd scare us. If we got close, he'd say, "Come here and knock on my wooden leg." Or when we first met him, he had a pocket knife. He's sitting there with his pocket knife, and he would jab it into his wooden leg and just freaked us out, just scared us to death. So anyway, we were kind of afraid of him, but he smoked like a chimney, one cigarette after another. And I remember I had learned, this was summer vacation, but I had learned the school year before from my teacher that they had just discovered back then that smoking causes lung cancer. And so I worked up the nerve, I remember, and I told that guy, I said, that smoking those cigarettes, you're going to get lung cancer from that. And he looked at me and he said, "Sonny, I've been smoking these things since I wasn't much older than you." And that created quite a conundrum in my mind because my teacher told us that smoking caused lung cancer. But this old guy says he's been smoking for a long, 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 long time, never has developed lung cancer. And I wonder if knowing what you know about predisposing genes to lung cancer and threshold effects, I wonder if you might be able to explain why it is that this old guy in his 80s who had been smoking for maybe close to 70 years, why he never developed lung cancer. And maybe this slide right here will jog your memory a little bit. We used this slide before when we were talking about the manifestation of neural tube defects in the developing embryo. But now let's talk not about that, but rather let's look at going from left to right on the x-axis, going from left to right, increasing numbers of predisposing genes to lung cancer, so that it looks like if you have this number of predisposing genes to lung cancer, represented by the red arrow, that would be the threshold where lung cancer would manifest itself. So, if you look at this situation right here, if a person was born with, let's say, this number of predisposing genes to lung cancer, would they develop, according to this chart, this graph, would they develop lung cancer? Think about that for a second. Okay, if you said no, you're right, because yeah, they've got a lot of predisposing genes that would predispose them to lung cancer, but yet the threshold is still to the right of that red arrow indicating the number of predisposing genes, so that person would be okay. But watch what happens to this particular person if we hit them with an environmental insult like cigarette smoking. Okay, watch what cigarette smoking could do to that person who has the number of predisposing genes to lung cancer indicated by the red arrow. Watch what, let's say, 5 years of smoking might do to that person. 5 years of smoking could move the threshold level back to that point, engulfing that, you might say, engulfing that red arrow, so this person would then develop lung cancer. Now, the problem is, you see, none of us knows how many predisposing genes to lung cancer we have. So tell me, let's go back to the old guy sitting outside the gas station, been smoking for 70 years, never developed lung cancer. I want you to think about the answer to this question. Explain why that old guy never developed lung cancer using this chart. Okay, if you said something like the following, then you're right. Maybe that old guy was born with, let's say, that number of predisposing genes right there where the red arrow is. Okay, so he's born with a very small number of predisposing genes that would put him at risk for developing lung cancer. Watch what 70 years, let's say, of smoking might do to that guy. Moves the threshold level back to here. He's still safe, see? He was lucky. He just managed to choose his genes carefully when he was conceived, which obviously is ridiculous, but you know what I'm talking about. The point is, the moral of the story is, none of us knows how many predisposing genes we have to disorders like lung cancer or alcoholism or depression or diabetes or any of these kinds of multifactorial conditions that you can think of. None of us knows how many predisposing genes we have to multifactorial defects like high blood pressure or high cholesterol. So what's the moral of the story? As we end this particular study guide right here, I'll let you consider that. I'll let you think about what do you think the moral of the story is. We'll see you later.